My name is Joanna Mountain, and I'm Senior Director of Research at 23andMe. I'd like to start by thanking the program committee for inviting me to take part in this conference, though I have to admit that I am very sad we can't be together to discuss this engaging topic of personal genomes. So I'm going to be talking about 23andMe's customer-powered research platform, but I'll start out by giving you a little bit of background of how I got to 23andMe. Starting with doing an undergraduate program in math and computational sciences, which I followed up with a stint with the Peace Corps teaching math to high school students in Kenya. Upon returning to California, a good friend known as DNA Dave Gilbert, introduced me to Professor Luca Cavalli-Sforza, with whom I worked for many years learning human evolutionary genetics. After graduating with my PhD, I moved on to Berkeley for a postdoc, and then back to Stanford to the Anthropological Sciences Department, where I had the opportunity to initiate a research program in Tanzania to study the speakers of the many linguistic families that are present in central Tanzania. But while I was at Stanford in about 2004, I read this phrase, Googling the genome, and I was really intrigued by that phrase, because as I was teaching about the genome, I thought, what if we could really just Google your own genome or somebody's genome and dive into it. So when 23andMe approached me in 2006, I was, very interested in this connection and the ability they wanted to provide of letting people look into their genomes. Though I did think they were very naive to believe that anyone would ever pay for that. But by 2007, I had decided I would give it a try, and here I am 14 years later, still at 23andMe. So I'll give you today a little bit of context about the personal genome service for those who are not familiar, and then dive into discussing the research platform and some of my favorite research. So 23andMe's mission is to help people access, understand, and benefit from the human genome. And the personal genome service is really all about accessing and understanding. So the way that works is somebody orders a kit via our website. They receive this tube, spit into the tube, package it back up, and send it to our partner lab. And then a few weeks later, they get to discover about themselves and their genetics through their personal, private, online account. And among the things they get to discover are, all via reports, including health predisposition reports, traits, wellness, and ancestry reports. And I'll say a little bit about some of those. So I'll start with a genetic health risk report. And these are my own results. And 23andMe estimates that I have a 13% chance of developing type 2 diabetes at some point between my current age and age 80. And that's, so that actually can be broken down by age. This chance, highest during my 50s and 60s, and then actually starts to drop in my 70s. So that's kind of fascinating. So this is the kind of access. This is I've got the access on the left here and the understanding on the right. But I will note here, I said this is the context, but this is really starting to get into 23andMe research, because this report is really powered by 23andMe customers, because it was developed using data from over 530,000 23andMe research participants. And that's it's kind of new for us to really power so many reports, using our customers' data. Now, 23andMe has a real focus on wellness and having something to do about your health, take action. So one of the things that the team has added recently is this option to put in information about yourself, age, weight, how many times I eat fast food, and how many times I exercise each week. I'm trying to be honest here. I don't eat fast food, and I exercise a few times a week. And that gives me roughly a 2%. That basically translates to about 2% of 23andMe research participants who have these characteristics currently have type 2 diabetes. Now I get to explore this. And on the right, what happens if I were to eat fast food every day of the week? Well, that 2% bumps up to 6%, just indicating that there's a difference between people, even if they have the same genetic characteristics and age and weight, that just that fast food diet can translate into a threefold difference in type 2 diabetes. Now take a step further and drop that exercise down to 0 times a week, keeping the fast food diet. And we go up to 9% of 23andMe research participants with those characteristics currently have type 2 diabetes. So in this way, I can sort of explore, like if I were to make changes to my diet, one way or the other, or my exercise, what the impact is in terms of my risk for diabetes. Of course, that's, me interpreting this as a geneticist, and I've got a lot of education to help me understand that. But for others, it's just an opportunity to kind of play around and get to know and really understand that we have ways we can, change our risk for type 2 diabetes. So a more traditional report is based on, 1 or 2 variants. And this is the case for our report on late onset Alzheimer's disease. And, this report looks at 1 variant in the ApoE gene. And now that's, can be a kind of a scary report for people. And so what we've provided, as you see over on the right here, a 3-minute video that is actually tailored to the result. If it said 1 variant detected or 2 variants detected, I would see different videos. And so I watched that video myself and been reassured, but not overly reassured, because we do worry about that. Now I took a look and explored the website further, partly because my mother had Alzheimer's before she died, and she also had no E4 variants. But I see here that, there's little chance of me developing Alzheimer's, by before age 65, but it does bump up a little bit. So the fact that I have no E4 variants doesn't mean I don't have any chance of getting Alzheimer's. It's still there, and that's demonstrated in this table. Because of that family history, I was curious to understand the non-genetic factors associated with Alzheimer's disease. And so I'm reading here, I get to read here about the diet, exercise, and intellectual activity. And one of the things mentioned here is fewer years of education have been associated with greater risk of developing Alzheimer's disease later in life. And given that my mother did not finish high school, maybe that's, maybe she's part of the training data set, or maybe, she did not have that cognitive stimulation early on. So we get into more lightweight reports in our traits section. Again, this is a report powered by 23andMe research. Over 840,000 23andMe research participants of European descent contributed to this study and this report. And I, of course, am actually one of the, probably one of the training data points, because I did answer that I prefer chocolate over vanilla. And of course it comes back and says, and that's not true for every report, because I'm just one of, 840,000 or close to 1 million when you take the data from all the different categories, the cohorts we have: African American, East Asian, European, and Hispanic, Latino. That gets much closer to 1 million. And just being 1 out of 1 million is not going to really influence the predictor. On the wellness side, we look at a number of other traits now. This again is powered by 23andMe research participants. And what I want to note here is that, going back to the Alzheimer's report, that was based on a single variant. Here we have a report for something, much less alarming maybe, but definitely something I have dealt with my whole life, cat allergies. And this report uses, over 11,000 genetic markers to actually give me my likelihood of developing a cat allergy, which puts me slightly above the typical range as seen in this diagram on the left. So I want to move now into my kind of favorite realm within 23andMe, which is the ancestry realm. Gets closer to population genetics. And I'll start just really give you an illustration via a friend of mine. I'm calling customer A, who's, this is his understanding of his family history. He identifies as African American, as did his parents and most of his grandparents. The one he understands was identified as Ashkenazi Jewish. But he also was aware of a couple of individuals who were known as Creek or Cherokee, and further back in his family tree. So when we look at his ancestry report, the main ancestry report, we see his understanding of his family history is really shows up here with 50%, over 50% sub-Saharan African ancestry in the pink and purple colors here. And you often see them on both chromosomes coming from both parents. The European is also, a large part of his ancestry, but that includes the Jewish ancestry, which is shown here in the green. Teal color, which is really, only on one of the chromosomes, because it only comes from his father. And the X doesn't have any of that teal color, because that is coming from his mother, who has a lot of the African ancestry. So the other thing that's shown here is the Native American ancestry in the yellow little patches that he understood come from mostly from his mother's side. Now, what is kind of cool is that we can look at the lengths of those segments he gets, traces back to each of his ancestries, and we can estimate the number of generations back to when he had that ancestor. And the Ashkenazi Jewish does look very recent, only going back a couple of generations, which fits with his family tree, being his grandfather. British and Irish goes back a little further in time, as do his sub-Saharan African ancestries, Ghanaian and Nigerian, go back really roughly 4 generations, which is very consistent with kind of the slave trade. And Native American goes back even further, to about 5 generations, which again is sort of consistent with what we see in his family tree. Now, I tend not to trust estimates to go back further than 5 generations, because those are based on very, very tiny fragments. But I really do trust these ones for the first 5 generations. So that's a great example. And I used, this customer, I used his case to kind of, validate what we're doing, which was we were certainly doing in the early years, trying to validate, because we had so many new customers and trying to understand if what we were telling them matched what they understood for themselves. Now, funnily enough, I'm jumping into privacy now, having just shown you the results of a customer. Well, I will just note that this customer has given us permission to share not only his results for health and ancestry, but even his name and his photograph, which I've have not included today, just to kind of be on the safe side. But privacy is a huge deal for 23andMe, and mostly we do not have permission, written permission from our customers to share their data. Mostly we don't. So we believe every customer has deserves a private and secure environment in order to access their genetic information. And to do that, we believe we need to be transparent about how our data practices, and so that they can make informed decisions. And we want to give them customers control of what they want to learn and what they want to share with whom. And we believe very strongly that it's our responsibility to safeguard and secure this customer data. And that's really served us well over the years. So what kind of information does 23andMe collect and process? It starts with the registration information, name and address, because we need to send the kit to somebody. the genetic information is core to everything we do. But the self-reported information is really important, especially for research. Now, web behavior information such as, a number of logins and which reports people have visited, we do collect. Those are mostly to improve the product. So yes, as I was saying, we use the data to provide our services, to improve those services, and most exciting for me, to conduct 23andMe research when customers have consented to that step. We've had to be very clear in ways we will not use personal information. We will not sell, lease, or rent that. We will not share data with any insurance company or employer. We won't provide that information to law enforcement unless required by law. And we've managed to avoid that so far. And we do not share customer data with any public databases without explicit permission from the customer to do so. And I mentioned control earlier. You have a number of ways we give the customer control over their data. They get to choose whether they store their sample. They get to opt into sensitive reports such as the Alzheimer's disease report. They get to choose whether they want to take part in research, whether they want to share information with others, whether they want to download their own data, and whether they want to delete their accounts, which does happen once in a while. So I've pulled from one of our papers our language around IRB review, just to make it very clear that our protocol has been reviewed and approved by an independent institutional review board called Ethical and Independent Review Services. And they've been really critical to us developing a strong research program. They review every modification that we make to our protocol, every new consent. And that is all about transparency. And I put here the link to our consent document, which is available at all times to both customers and non-customers. Again, this is about being transparent. All right, so let me get into the research. And the part of our mission that's about research is the benefit from the human genome. We believe that through our research, we can enable many people to benefit from the human genome. And so we have an interconnected business, and these are the two main elements I'm going to discuss now. We have the customer's perspective. For them, the personal genome service provides an opportunity to learn about their own genetics, ancestry, and health. It also gives them an easy way to participate in research. From the research perspective, we are able to recruit consenting participants from diverse ancestries and those who have diseases of interest. And once we do that, our team can publish papers as well as generate new reports for customers. So that's the research part of it. And they really support each other, these two arms of the business. So our research platform is really this simple equation. We have these consenting customers who consent that we can use their genetics, and they respond to surveys. And we pull those all together to make discoveries. Now, there are a number of advantages of this online research. The participation is fairly easy. Geography is certainly not a barrier. Anyone who has an internet connection can participate. And most people end up taking part in multiple studies. And we actually let people know how many publications they've contributed to in one part of the website. And at one point, I had contributed to 74. I think it's even higher now. So some of the principles behind our research platform, customers consent to participate and actively opt in rather than opt out, which is we see as critical. The research is overseen by an independent third-party IRB, as I mentioned. Customers can withdraw from research participation at any time. And when we release data, it's in an aggregated and de-identified form. Really, we release summary statistics when we publish, for instance. So at this point, we've sold over 12 million kits. About roughly 80% of those individuals consent to take part in research, which is thrilling for us. And they not only consent to take part in research, but they respond to surveys. So we have, over 3 billion responses to survey questions at this point. Very powerful database. Now, I will note that like many large genetic databases, we have an overrepresentation of people of European ancestry at 73%. That is something that we are very aware of, and we're always trying to increase the proportions of these non-European cohorts. In terms of sex and age, we have broad distribution of ages with kind of large cohorts in the 30s and 60s. At this point, we have, over time, it's roughly 50% male and female, but at this point, we're slightly enriched for female participants. So each person contributes to an average of 200 genetic insights. And with those contributions, we've been able to publish well over 150 publications, peer-reviewed publications, and that includes 1,500 genetic discoveries, because there's more than one per paper. But this is all founded on this very large database. I mean, that's what it's taken. And that was the vision early on. Anne had this vision that we need to get beyond 1,000 cases and 1,000 controls. That's not getting us anywhere for conditions such as depression. And so what we have is in all these clinical areas, we have customers with these a wide variety of conditions. And I mentioned the ApoE gene, the E4 carriers. We have well over 1 million of them. Another over 1 million have reported depression, and it goes on. So we have, and even down to the rare diseases. They may be rare, but we've been able to identify thousands of cases. Now, this is in some cases we've really made an effort to recruit rapidly, and that's the case for our Parkinson's disease cohort where we enrolled 11,000 individuals in just 4 years. If we go to the depression and bipolar cohort, that we enrolled 25,000 individuals in just 14 weeks. So we have the ability to recruit fairly rapidly, especially by working with advocacy groups. So let me get into some of my favorite studies. So this study of depression that we published in 2019 is certainly a collaboration with external academic groups, but it's a meta-analysis of over 800,000 individuals. It's certainly the largest depression study to date at that point. And the cool thing was there had been many studies, GWAS studies of depression that did not really yield strong results. But here we were able to find 102 independent genetic variants associated with 269 genes, including 15 gene sets. So that's the exciting part of that paper. Now, here we look at circadian rhythm, and this yielded over 350 SNPs associated with circadian rhythm, using data from close to 700,000 individuals. And the cool thing that's added in here is we use Mendelian randomization to examine causality. And this was looking at schizophrenia and circadian rhythm, the causality relationship there. This is going to a completely different topic of spontaneous preterm birth. And previous studies had suggested that there was quite a bit of genetic based risk for preterm birth. And so this study really was looking at variants in this one gene, HSPA1L, and we were able to contribute GWAS data and find this association was strong. So really was combining the exome sequencing done by an external group and our genotyping. We're really able to solidify the role of the variants in this particular gene. So, penetrance has always been a challenge. And, this PRNP gene, prion disease gene, was thought to be pathogenic, or many of the variants in it were thought to be pathogenic. But what we found was these so-called pathogenic variants were much more common than had been previously understood. So many of the variants turn out to be benign, others have incomplete penetrance. And so this research changed prognoses and also provided therapeutic clues. So there's all kinds of ways that exploring a large database can lead to biological insights. So our aim is to make discoveries, share those with the customer, and ideally create an educated, engaged customer cohort, and then they will contribute to making further discoveries over time, because we continually invite customers to answer new questions or to update their health status in our database. So I mentioned the publications, and we have a page that lists all of our publications, and you can even filter by topic. The most recent one listed here is one on skin cancer, led by Pierre Fontanillas from our research team, published in January of this year. But all these publications, as I noted at the very top here, we make sure that they're available open access, because imagine we're using data from these customers to publish papers, and if they couldn't get access, that just seemed wrong to us. So from the very beginning, we've made sure that we publish in open access journals, or we pay to make a particular publication available to anyone who's interested. It's certainly very helpful to me as I go and explore the papers. I always know that I can go and find it without having to go through some paywall. So some of these papers I mentioned were collaborative. Many of them actually were, the ones I mentioned. And a few of those came through our Research Innovation Collaborations program, where we invite academics to submit proposals to team up with us and examining our own data in parallel with their data. So the next deadline for that program is in July of this year. When we publish, we often we make the data sets available to qualified researchers through this data access program. And I'll come back to the Populations Collaborations program a bit later on, but that deadline is next fall. So I mentioned the two components of our business. We have our personal genome service that goes straight to the customer. We have our research program. And since 2015, we've had a therapeutics division, which is leveraging data to accelerate data discovery. And the goal is to address unmet medical needs and rare conditions. So a bit more about the therapeutics program. And this was inspired by, the knowledge that, that we were able to demonstrate, as have others, that drugs with human genetic support are 2- or 3-fold more likely to succeed. And our scientists can use genetic associations to develop better, more personalized treatments for serious unmet medical needs. So that's the mission of the therapeutics division. And just to show how that works, we put together the survey and genetic data, run the genome-wide association studies and phenome-wide association studies to identify a target, a molecular target. And once we have that target, a lot of biology happens in order to discover a drug that might interact with that target. And then the really promising step is when we get to the clinical trial phase. And the goal is to have product approval of one of these drugs. And so you can run many programs at once through this pipeline. And there's one that's in the clinical trial phase at the moment, a cancer therapy medication. So it's an exciting component of 23andMe's business is the therapeutics division. So something that's been important to me the whole time I've been at 23andMe, since the day I started, was the question of the diversity of the database. I mean, of course, that's what I've been fascinated by as a graduate student and postdoc. So I've been involved in many initiatives at 23andMe to promote diversity in research. And I'll just point out two here. We've done 2 sequencing projects where we sequenced over 1,000 individuals, 1,000 African Americans, 1,000 individuals who are Native American or Latinx. And these projects enable us to build imputation panels that improve the imputation quality for individuals who have either African or Native American ancestry. So I've been very happy that we've been successful with those projects. Now, in 2011, acknowledging that very few participants in genetic studies were of non-European ancestry, we kicked off a program called Roots into the Future, where we invited 10,000 African Americans to take part in research and become 23andMe customers. Fortunately, things have changed a little bit since then. The overall published fractions of non-European research participants in genetic studies is closer to 10%, but we still have a long way to go to get to the sort of census fractions that we would like. I've been championing a project called the Global Genetics Project, where we invite people from countries that are underrepresented in our database to sign up for 23andMe. And we say your participation in the Global Genetics Project will help diversify our database and boost studies in populations that are underrepresented in genomics research. You'll learn more about your heritage and help others around the world discover theirs. So this is an exciting project, and we've enrolled close to 10,000 individuals from across the world, and including individuals who have recent African roots, because I have long wanted to examine the part of this story here. This is the triangle of trade, between 1550 and 1865, with this one pink path here being the slave, the forced deportation of slaves from Atlantic Africa to the Americas. So I'd long wanted to look at that. And, what I found was maybe about 2 years ago, we had data from across the Americas of people with African ancestry, and we had pretty good representation all along Atlantic Africa, with the efforts we had made. And so I was excited to sort of see, can we look at the genetics and do we see the same pattern that we see in the historical records? And the wonderful thing was, at about the same time, many of these manifests, these shipping manifests, had been sort of put into a database called slavevoyages.org by David Elsis and colleagues. So we were able to pair up the genetic data and the records data. So the records data are here, the embarkation counts leaving Atlantic Africa, and the disembarkation counts arrivals in the Americas. Now, not everybody is represented in our study or genetically, because about 2 million people died in this passage, this middle passage. So they are not represented in the genetics. So we had many expectations as we went into the study, and I'll just mention one of them. We expected that regions that had more enslaved people disembarking in them in the Americas would have more African ancestry. But we found something contrasting to that expectation. We actually saw the highest African ancestry in the United States and the British Caribbean here. And that was not what we expected, and much lower African ancestry in Brazil and South and Central America, which is where most of the slaves went. And so when we discussed this with our historian advisors, they pointed us to literature on various practices and ideologies across the Americas. And in Brazil, there was a practice of basically not worrying about a slave dying because they were easily replaceable. So many slaves were being brought, especially from Southwest Africa to Brazil, that there was very high mortality for that reason. Whereas in the US, once slavery had been, or slave trade had been abolished, there was sort of, an effort to maintain slaves by forced reproduction, which would have increased the African ancestry amongst those slaves. So it really kind of pointed us towards a kind of horrifying part of the history, as we saw the discordances between the shipping records and the genetic patterns. Though we did see overall agreement, the general broad pattern of who left Africa and who shows up in the ancestry of peoples in the Americas is very clear. But when we do see discordances, they were tended to be consistent with independent historical accounts of, as I mentioned, mortality, rape, national ideologies, and other poor treatment of enslaved people. So it's kind of amazing that this shows up in the genetics of people living today. And I'll point you, again, this is open access, as all of our publications are, to this paper we published last year in the American Journal of Human Genetics. I encourage you to take a look at that. So with that, I'm going to wrap up and thank the millions of 23andMe research participants who've contributed to the studies I've mentioned today, as well as many other studies, and also thank my colleagues at 23andMe in all the various corners of the company. It's been an exciting research journey, and thank you for listening. I hope we get to have a conversation about this sometime. Bye-bye.